Hello and welcome to my channel. In this lecture we'll conclude our discussion of the blood elements, and we will talk about plasma and then lastly the lymph. So let's begin. Platelets, it's one of the blood elements. Besides the RBCs and white BCs, they are called thrombocytes, and they circulate the blood with their function is clumping reaction in case of any blood vessel energy. They form the blood clot. The platelets have no cell nucleus. Actually, they are fragments from a bigger cell called the megakaryocyte present in the bone marrow, and you can see a couple of megakaryocytes here in the bone marrow, and after that, after being released from the megakaryocyte, it enters the bloodstream, where it appears on a blood smear as dark spots, as you can see here. These are the platelets. The inactivated platelets means that not clumping. They are biconvex discs. Means lens-shaped, and when activated means in the formation of a blood clot, they have cell projections or cell membrane projections covering their surface. On a stained blood smear, like you see here, they appear as dark purple spots, as mentioned before, and they are less in diameter from the RBC. Of course, RBC is more abundant than platelets, and the one major function of the platelets is hemostasis. Hemostasis means the process of stopping the bleeding in case of injury. Low platelet concentration is called thrombocytopenia, either due to decreased production or over destruction or increased destruction in cases of hypersplenism, for example. Elevated platelet concentration is called thrombocytosis, and this may be congenital or due to myeloproliferative neoplasms or other certain or other myeloid neoplasms affecting the the cells, the precursor cells of the producing the blood elements. Normal platelets respond to any abnormality on the vessel wall. Sometimes clotting appears or happens in an intact vessel because the inside of the vessel is affected by an irregularity, such as atheroma, for example, in atherosclerosis. And so clumps may be produced in veins producing venous thrombosis, arteries, arterial thrombosis, or microcirculatory thrombosis. And in case of an artery thrombosis, this leads to narrowing of the artery lumen, means ischemia of the tissue supplied, and if the narrowing continues and leads to obstruction, it leads to the death of the tissue supplied by such an artery. The development of platelets from totipotent or immature stem cells in the bone marrow and develops into a megakaryocyte under the effect of a hormone or a substance called the thrombopoietin, a hormone produced in the kidneys and the liver. Each megakaryocyte gives or produces 1,000 to 3,000 platelets during its lifetime, and there is about 10 up to the power of 11 platelets produced daily. There is a reserve plates stored in the spleen and released when the body needs it by the splenic capsule, um, 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 induced by sympathetic nervous system. The average lifespan of the platelets is 8 to 9 days, and old platelets are destroyed by phagocytosis in the spleen or the liver. Now we're done with platelets. Let's talk about plasma, the fluid media that the blood elements suspended in. It's a yellowish liquid component of blood. It's the intravascular part of the extracellular fluid, and it's mostly water. Contains important dissolved proteins like albumins, globulins, and fibrinogen, glucose, clotting factors, electrolytes, hormones, carbon dioxide, which is the main medium for as an excretion of the carbon dioxide from the body. The plasma plays a vital role in maintaining the osmotic pressure of the concentration balance in the circulation, and the osmotic pressure is one of the driving forces at the capillary bed, for exchange of material, and the other being the hydrostatic pressure of the blood itself. It protects also body from infection. A blood serum is blood plasma without the clotting factors, and the term plasmapheresis is for medical therapy, a kind of medical therapy where plasma is extracted and treated and reintegrated in the body, and you can see here a device for plasma pheresis. The proteins present in plasma are three different kinds of proteins. The main proteins are albumins, globulins, and fibrinogen. Albumin is the most common plasma protein, and it's the one responsible for maintaining the viscosity and osmotic pressure of the blood. The viscosity of the plasma prevents the fluid from entering the bloodstream from outside the capillaries. Albumin is produced in the liver, so its levels are affected in liver diseases. Globulins, the second most common type of proteins, they constitute or form the immunoglobulins, which are antibodies essential for the immune response, and also transport globulins like the TBG or the thyroxine binding globulin. They transport hormones in the blood by binding to them and then releasing them at the target organs. Also, there is fibrinogen, responsible for the clotting of blood to help prevent blood loss. So we have albumins, globulins, and fibrinogen. The color of the plasma is normally yellow due to bilirubin, presence of bilirubin, carotenoids, hemoglobin, and transferrin. The change in color, it may be green color due to presence of ceruloplasmin or sulfhemoglobin, maybe dark brown or reddish in color in cases of hemolysis, in which there's methemoglobin levels are released from the broken blood cells. The plasma is normally transparent, but sometimes it's opaque. The opaqueness is due to elevated content of lipids like cholesterol and triglycerides. Lymph, the last of the body, the fluid tissues we were talking about. The lymph is composed of is a fluid that is a collection of the interstitial fluid, the excess that is left over after the exchange of blood at exchange of that happens at the capillary bed, collected as lymph, and propagated in lymph vessels that passes into intervening lymph nodes that process the lymph running into them. Then eventually the lymph is returned back to the venous system through the subclavian veins and the major lymph ducts on the left side, thoracic duct, on the right side, the right lymphatic duct. It collects the interstitial fluid between the cells of the body tissues and propagated to lymph nodes, where substances are removed by tissue lymphocytes, and circulating lymphocytes are added to the fluid before emptying eventually in the venous system. There's a continuous exchange at the capillary bed, so the structure of lymph is variable, is changeable. It's changing. Its composition continually changes because of the continued exchange at the capillary bed. It's similar to blood plasma. Returns proteins and excess interstitial fluid to the bloodstream, and also responsible for transport of fats from the digestive system in the form of chylomicrons. It's the major pathway for absorption of fats from the intestine. Also, it may contain bacteria that eventually will be destroyed at the lymph nodes, and it's a channel for spread of metastatic cancer cells that might be transported via lymph. That's why in removal of cancer, you remove the associated lymph nodes as well. The structure of lymph, similar to that of plasma. It leaves the lymph node richer in lymphocytes, and the lymph coming from the digestive system, called chyle, it's milky white because of its high lipid content, and you can see it here, milky white appearance of the lymph. Functions of lymph. Lymph returns proteins and excess interstitial fluid to the bloodstream. It picks up bacteria, transports them to the lymph node where they are destroyed. It's a pathway, it's a channel for spread of metastatic cancer cells, and also transports fat from the digestive system, beginning in what's so-called the lacteals, the blind end lymphatic channels present at the intestine, to the blood via the chylomicrons. I hope you got the maximum benefit from this lecture, and thank you very much for watching.